Well, good morning. I'm happy to see how many of you were able to swim in today. It's quite a rain last night. Who didn't get any coffee this morning is about to pass out fast. If you respond that quickly, I doubt you need the coffee that much, but what the heck. And who could use a sardine? You still interested? In that case, you get the sticky bun. Okay. Could you pass that back to that ravenous person? Okay, we're going to have a lot of fun today. Before I forget, I have to tell you, I have to cancel Friday's office hour because I have to be at a meeting in San Diego by about noon on Friday, so I'll be dashing out right after lecture. I was able to get a room in Stanley tomorrow, room 221 from 2 to 3, so I'll have a make-up office hour tomorrow, Stanley, 2 to 3, room 221, if anybody can make it. We're going to have a lot of fun today, although there's going to be a risk that it's going to give you a headache and make your head spin for a while, especially if you didn't do the reading before coming to lecture. Because what I'm going to do today is to give you a sense about why it is that I think it's so important that you know so many different things. And the reason is that progress in biology doesn't really boil down to putting together a few fundamental equations that describe the physical universe the way physics has the pleasure of. Rather, in biology, what we have to deal with is the nature of evolution having put together things that which are good enough here and close enough there, and we need to understand the nuances and the specialized situations in different organisms to be able to exploit those, to do experiments that otherwise would be really quite difficult. And the other thing about biology that's really sort of fun is that the biggest advances come when you're able to put together things that you would think were not related at all. And as an illustration of that, today we are going to have stories from the following different areas. We will start our stories with studies in mammalian cells and culture. We will then take a tour through experiments on how frog egg development can be activated. We'll do a little clam biochemistry, transition seamlessly into yeast genetics, a little bioinformatics, peptide chemistry, and end up with 5 Nobel Prizes by 9 o'clock. So that's where we're going. Now, all of these different threads of logic will be used to try to understand the eukaryotic cell cycle. Last time, we had a lot of fun talking about the mechanisms by which cells divide in mitosis. And we spent a lot of time thinking about the idiosyncrasies of the spindle and the chromosomes and the kinetochore. But today we're going to be dealing with cell division at the executive level. How is it decided by the cell when it's going to divide? And the big picture issue here will be to ask whether cell division is driven by a clock inside the cell, such that at 2 o'clock S phase happens, it's over with by 4, and at 8 o'clock mitosis happens, or is there some kind of regulated internal oscillator that drives cell division? And the answer will be that there is not a clock, and by the end of today you'll understand how this oscillator works. So that's where we're going. Now I want to start with an experiment, and I hate to admit it, I had no idea how significant this experiment was when it was first done. In fact, I didn't even read it when it was first done. I didn't even read this paper until 3 years after it was published. But it has turned out over time to have been an incredibly influential experiment. And I want to share it with you as an example of how taking the time to really understand what's happening in literature can really, really be useful. So this is an experiment that was done by a team called Rao and Johnson. It was published in 1970 in a journal called Nature. And what they did was to try to ask what the rules were about how cells were controlled in different parts of the cell cycle. Now they used a trick, a virus that was discovered in Japan, in Sendai, Japan, called the Sendai virus. And what they were able to do was to take cells that were in different phases of the cell cycle and fuse them together by adding this virus. It causes the membranes to melt together. And then they looked at that fused cell, which goes by the name heterokaryon. That name is not important, but you'll probably see it in your text at different places. It just simply means a fusion of 2 cells. And they asked, what happens? So here's an experiment. If you take a cell that is in M phase and you fuse it to another cell that is in G2, in that heterokaryon, the G2 nucleus immediately goes into mitosis. Okay? Now, is that interesting? It's not interesting until you do the control. That is, if you take a G2 cell and fuse it to a G2 cell, and that heterokaryon, the two G2 nuclei, stay in G2 for some period of time. Eventually, they go into mitosis. So with the benefit of that control, we're now prepared to say that there is something active in this cell that can drive this nucleus ahead into the cell cycle at a time that's premature from what it would have done so ordinarily. So that's the sort of the logic that we apply to this kind of experiment. Does everybody see that result and that interpretation? Because of this control, we know that this nucleus has something, the cell has something in it that is driving mitosis because it can drive this cell into mitosis prematurely. Any questions on the logic there? Okay. Let's go on. We'll have some more fun. Here's an interesting one. Let's take a cell that is in S phase and fuse that cell to another cell that is in G1. Okay? What we see in this fusion is that in the heterokaryon, the nucleus that was in G1 immediately begins DNA replication. So the S phase nucleus induces DNA replication in the G1 nucleus. Now, that by itself doesn't sound all that interesting. After all, we discussed earlier all of the enzymes that are required to make DNA replication happen: DNA polymerase, DNA ligase, primase, helicases, and all of that. And if those enzymes are in this cell active and you fuse them, why wouldn't the enzymes go into that nucleus to activate it? So that's not so surprising until we do the next experiment. Let's take a cell that is in S phase, so making DNA left and right, fuse that to a cell that has already finished replicating its genome. That cell has a nucleus that's in G2. And what we find is that the S phase nucleus cannot activate replication in the G2 nucleus. Okay? Instead, what happens is that this S phase nucleus finishes S phase, catches up to G2, and then both nuclei synchronously go into mitosis. Okay? So, and the other important point is that the G2 nucleus, which would have gone into mitosis earlier, does not go into mitosis because the S phase nucleus is retarding that. Now that's a sophisticated result, because we know that all the enzymology necessary to replicate DNA is in this cell. This nucleus is replicating. That one is not. And we know that this one is poised to go into mitosis, but it does not because of a signal from this cell. So this very subtle but beautiful experiment was really one of the very first pieces of evidence for something that has become a very big deal in cell biology called checkpoints. And I like to think of checkpoints in a sort of a generic sense as any mechanism that retards step n+1 from occurring until the previous step, step n, is complete. So there is a checkpoint here that is blocking this cell from going forward until S phase is complete. Okay? There is also something very interesting about that nucleus that is blocking its ability to initiate its DNA replication. And alas, we probably won't have time to talk about that in this course, but in MCB 110, you'll definitely get details about that. And for the poor soul who said that they weren't planning on taking MCB 110, maybe you should reconsider. Any questions on the phenomenology here? By the end of today, you're going to understand how checkpoints work to control the cell cycle. Questions? Okay, so that's chapter 1. For chapter 2, we're going to discuss the work of a Japanese scientist named Masui, who is extremely influential in cell biology, with his discovery of something called the maturation promoting factor. This is a really obscure name for a very interesting molecule, so let me explain how it's got such an obscure name. One of the favorite organisms for cell biologists is the African clawed toad, Xenopus. And the Xenopus mamas make oocytes, which are immature eggs that can be squeezed out of the abdomens of the mama oocytes with no harm to the Xenopus, but for the delight of experimentalists. They're about 1 millimeter or so across, so they're not so big, but under a low-power microscope, you can do remarkable things with them. Now, this Xenopus oocyte, as it is squeezed out of the mama frog, is arrested in metaphase 2 of meiosis. Now, you heard about meiosis 1 and meiosis 2 in high school. We'll get to meiosis in detail on Friday, but just remember that metaphase 2 of meiosis is essentially a cell that is poised for a mitotic division, because the second meiotic division is in reality a mitotic division. Now, when this cell, this oocyte, is ready to develop, the metaphase 2 arrest is released, the cell goes through a mitosis, and you can see that by simply observing the appearance of a white or light-colored spot on the dark-colored hemisphere of the oocyte. All right? So this is an assay for the activation of mitosis. You simply look for white spots on dark hemispheres. And this appearance of this white spot is called maturation, the onset of meiosis 2, which is in fact a mitotic division. So what Masui wanted to understand is, how is it that the cell cycle is activated? What drives mitosis? And he took advantage of experiments that others had done who showed that if we take an oocyte at this stage and inject progesterone, a maturation hormone, that certain proteins get made that cause the cell to become activated. So we would see a white spot on the hemisphere because what's happening is mitosis has become activated. Okay? That was an observation by somebody else. So what Masui did was then to take a syringe, stick the syringe into this oocyte, and now rather than inject progesterone, actually pulled out about 1 nanoliter of the stuff in that oocyte. Okay? So now the experiment that he asked was, is there an activity in this oocyte that is driving mitosis, and what is it? And he then injected that into a new oocyte that had never seen progesterone. He blocked protein synthesis by using an inhibitor called cyclohexamide. And what did he observe? The onset of mitosis. Okay? So that's an important result. That tells us that there is something which is active here in the cell that is actively going through mitosis, that when injected into a cell that is not active in mitosis suddenly becomes active in mitosis. And it's an activity that does not require de novo synthesis, because cyclohexamide would have blocked that. So what is that? Well, Masui was worried that maybe it was a little bit of progesterone, maybe it was a little bit of something that was carried over. So he took this activated oocyte and used his syringe to suck out another nanoliter from that one, and used that to inject yet another oocyte, a naive oocyte that had never been involved in the experiment before, injected it, and bango, mitosis happened again. And he did this experiment again and again and again. And he expected, if there was an activity in this cell that was activating mitosis, that he might be able to dilute away that activity. So after enough rounds of sucking and injection, it would no longer be active. But he found that this activity was not dilutable. Therefore, he deduced that it must be an autocatalytic activity that was maturing the oocytes, driving mitosis, and that autocatalytic activity he named MPF. And the autocatalytic activity, its non-dilutability, implied to Masui that the MPF was in fact an enzyme. So I want to pause and see if people understand the MPF assay and the logic about the autocatalytic nature of this enzyme. Any questions about that? Yes? So the question is, what does it mean to be non-dilutable? And what that means is that no matter how many times you suck an activity out of an activated oocyte and re-inject it into a naive oocyte, when that oocyte becomes active, you can continuously do this experiment time and time and time again without ever losing that activity. Okay? Yes, question in the front.
I didn't quite catch it, but what exactly is the spot that forms?
The spot, well, it's an indication of the second mitotic division, and really what it is it's a series of vesicles that appear and displace some of the yolk, the dark-colored yolk on the oocyte. It's just a way of, by looking at an oocyte, knowing whether or not the second meiotic division has occurred. Okay? It's the assay. Yes, question there.
What could it have been besides an enzyme?
Ah, well, what could it have been beside an enzyme? Could have been the product of an enzyme, right? An enzyme might have made a molecule, right, that, another hormone, for example, that caused this activity, and if a limited amount of that was made, then as you continuously do these cycles of extraction and reinjection, you would dilute that product. Okay? So this is an enzyme that has to be able to activate itself. Is there a question there? Yes.
So when you're syringing out the cell, is it syringing from the cytoplasm or the genetic material itself, or all of it?
Oh, the question is, what are we sucking out? Yeah, so there is no distinction between the nucleus and the cytoplasm at this point because the nuclear envelope has broken down. And so we're just sucking out a small fraction of the juice inside the cell, the plasm. Okay? Yes, in the front.
Even though it's an enzyme, but after rounds and rounds, wouldn't we be sucking out less and less enzyme so the concentration is diluted?
So the question is, shouldn't the concentration of that enzyme be diluted with many rounds of sucking out and reinjection? That was the expectation. But the observation, the miracle, so to speak, is that that doesn't happen. It doesn't get diluted away. So what you should be thinking then is, so what must be going on? There must be some kind of a precursor form of this enzyme that's there but not active. But once the enzyme is active, it can continuously reactivate that precursor form. That's the kind of thing that's got to be going on in the back of our minds. Okay? So Masui and those in this field came up with this hypothesis then that in the cell cycle, the level of the MPF activity would be low in interphase, high in mitosis, low in interphase, high in mitosis, low in interphase, high in mitosis. Now you might think that this experiment's just coming from frog eggs and that it might not be applicable to other things, but Masui really clearly understood that what he was studying was a fundamental aspect of the cell cycle, and he just took advantage of the frog egg as a way of getting something big and easy and biochemically tractable. He hoped to be able to look at mitosis, even though the context of that mitosis was the end of a meiotic cell division. So this is taking advantage of the idiosyncrasies of biology. Are we ready to move on to clams? Okay. So one not very good prediction of Masui's work and others is that there would be a protein that would be synthesized each M phase, and that protein would drive mitosis. This hypothesis overlooks the autocatalytic activity of MPF, but this was the hypothesis that was driving people's thinking at the time, and probably for not particularly good reasons. Tim Hunt, a very good biochemist in Cambridge, England, decided he was going to look specifically for proteins that were synthesized at particular periods in the cell cycle. Now, he chose for his experiments to use clam embryos. The reason he chose clam embryos is that there's a kind of a synchronous development early on in clam embryos, spiral cleavage, in which the cell cycles are incredibly synchronized. So if you start with a fertilized clam egg, you can have more than 10 rounds of synchronous divisions. So you'd have 1,000 cells per fertilized egg, each of them synchronously going from interphase into M phase to interphase to M phase. And because clam embryos are cheap and abundant, you could get a zillion of them. And if you're going to do biochemistry, you need lots of material, so zillions of embryos is a good thing. You could synchronize the fertilization and hence have zillions of synchronized cells going through mitosis. So to look for proteins that were synchronously made in the cell cycle, he simply used protein gels, like you've seen in the text, to look for in these protein gels bands representing a particular species of a protein that became more abundant in one part of the cell cycle than the other. And so in blue on this graph is a representation of some such protein. He found a protein whose level increased consistently through interphase and into M phase, and then in M phase, the level of that protein dropped precipitously. In the next cell cycle, it rose, and then it dropped precipitously. And in the next cell cycle, it rose and dropped precipitously. This was a protein whose level cycled in the cell, and with typical British creativity, he named this protein cyclin after its ability to cycle. Okay. Then he did a very interesting experiment. Using slightly more sophisticated methods, he asked, what is the rate at which that protein is synthesized? I think he expected to find that it was synthesized at an ever-increasing rate in the cell cycle, but instead, what he discovered is that it is synthesized at a continuous, constant rate throughout the cell cycle. Okay? So we have two results here that don't easily fit together. We have a protein that is made at a constant rate, but its level cycles. What could be the explanation for that? How do you get cycling levels of a protein that is synthesized at a constant rate? Anybody want to take a crack at that? Yeah, what's your name? Pat, what do you think?
Probably have some other something in the cell that's breaking it down at various rates, maybe.
Pat's hypothesis there is that there must be something that is periodically destroying the protein. So that if we're making it at a constant rate and it accumulates, suddenly here something becomes active that destroys that protein, which will turn out to be exactly the answer to that. That's great. Okay? So the idea then is that this protein gets to a certain level, crosses some threshold, and that threshold then may be important for activating mitosis. Now, Hunt did a number of experiments and discovered multiple cyclins, multiple proteins whose abundant cycles in the cell cycle, but different cyclins peak at different points in the cell cycle. Cyclin B peaks at M phase. He found another cyclin called cyclin A that peaks at S phase. Okay? So these are very interesting proteins then, whose activity cycles, whose level cycles despite a constant rate of synthesis, and the time at which they peak is interesting with respect to big events in the cell cycle. But there was no definitive experiment that said that the cyclins were responsible for driving the cell cycle. They may have been, their levels might have been responding to other things in the cell cycle that were not determinative of the cell cycle. So questions on cyclins. Clam biochemistry simply because it gave him the opportunity to have large amounts of extract from cells that were synchronously going through mitosis. Any questions on that? Okay. We're going to switch to yeast now. You've seen yeast in the lab in terms of complementation experiments, but I'll tell you about experiments that were done in two different labs, the lab of Lee Hartwell and the lab of Paul Nurse. These two people are among the best yeast geneticists in the world, and one of our very own colleagues, Doug Koshland, who I see in the audience today, actually trained in Lee Hartwell's lab during the time of some of these experiments. Studies of yeast genetics have allowed us to understand a lot about how the yeast cell cycle and ultimately all cell cycles are regulated. And to give you a sense of the yeast cell cycle, I'll show you a diagram of Saccharomyces cerevisiae, one of the yeasts that is used for these experiments. Yeast in G1 are recognizably in G1 because they lack the bud. So yeast have a mitosis like human cells, but unlike human cells, they have an asymmetric division of the cell. So yeast divide asymmetrically by budding rather than symmetrically in cytokinesis. S phase is indicated by the emergence of the first bud on a cell, and the cell's bud gets larger as the cell goes through the cell cycle. M phase is recognized by a large bud with a nucleus stretched between the cell cycle, and cytokinesis is represented by the bud achieving a certain size and having an intact nucleus within it. So simply by observing a yeast cell and where that cell is, observing the yeast cell, you can tell where that cell is in the cell cycle. Now, if we want to study the cell cycle genetically, we need to find mutations that disrupt critical points in the cell cycle. Now, the cell cycle is, of course, essential for life. So how do you have mutants in proteins that are essential for life in a way that you can still study the cells? And the answer is to use conditional mutants. So let's consider Joe Random protein, whose structure is represented by this knot. Okay? Typical proteins at 23 degrees will have the same structure as they will have at 37 degrees if they are as nature made them. However, if we have a temperature-sensitive mutation in the gene for a particular protein, that protein will lose, let's say, a kcal of free energy for folding, and in such mutants, the protein will be functional at the low temperature, but if we shift the cells to the high temperature, the protein becomes denatured, unstable, aggregated, and destroyed. Okay? So the use of temperature-sensitive mutations allow us to find genetic blocks in particular proteins, yet we can grow the cells with such mutant proteins under one condition and evaluate their phenotype, their defect, under other conditions. So what Hartwell and Nurse both did was to isolate very large collections of temperature-sensitive mutations in two different yeasts, Saccharomyces cerevisiae and Schizosaccharomyces pombe. And they both discovered genes that they named CDC genes for cell division cycle genes. And because of the perverse nature of luck in the universe, the gene in Cerevisiae, called CDC28, is Cerevisiae's version of the same gene that by chance was called CDC2 in pombe. And this is one of these nomenclatural annoyances that we have to deal with. So this is going to become a very important gene and a very important protein, and they go by both of these names. We just have to deal with it. So let's pause for a second to think about, well, how do you find these mutants? Well, it's really pretty easy. We take a culture of cells, and we bathe them in a mutagen, like ethylmethane sulfonate, an alkylating agent which changes bases here and there. We take these mutagenized cells out and simply plate them onto a Petri plate of rich media, and we incubate that Petri plate at 23 degrees, room temperature, and we let those cells grow into colonies. Now, some of these cells will have suffered a mutation which is a non-conditional mutation destroying an essential gene, those cells on this plate we will never see because they're dead. They can't make a colony. So we look then only at the cells which have no complete defects in any essential gene, and we take this Petri plate and simply replicate these colonies onto another plate that we incubate at 37 degrees. And so what we're able to do then is to ask, is this mutant temperature sensitive? No, it's growing on this plate. Is that mutant temperature sensitive? No, it turns out only this mutant is temperature sensitive. Okay. So that's how one finds temperature sensitive mutations in essential genes. Hartwell and Nurse both collected hundreds of such temperature-sensitive mutations. They sorted them into different genes by the complementation test that you've seen already in the lab. And among the genes that they discovered, the CDC28 and CDC2 gene turned out to be the most important for our discussion today. So what's the characteristic of these mutants that make them so interesting? If we take a wild-type strain of yeast and isolate a single cell at 23 degrees and move that cell to 37 degrees, it makes a whole population of cells at different phases of the cell cycle. If we take a budded cell, move that to 37 degrees, it too makes a lot of cells. But the gene that's really quite interesting here, CDC2TS, if we take that mutant as a single unbudded cell at 23 degrees and shift it to 37 degrees, what we see is that nothing happens. It stays right there in G1. But if we take a cell that has started S phase, and we can tell that because it has this little bud on it, that cell will then complete the cell division cycle and arrest in G1. And if we take a cell that is in M phase, move it to 37 degrees, it too arrests in G1, having completed the cell cycle. So this tells us then that CDC2 or CDC28 ordinarily encodes a protein that is required to initiate the cell cycle, but it is not required in any other phase of the cell cycle. Once it is initiated, it can continue to go through that cell cycle until it gets back to G1 again. So this is a protein that is specifically required in the initiation of the cell cycle. Okay. Any questions on that? So far, you should not see any obvious connection between clam biochemistry, frog eggs, or mammalian cells in culture. They should seem to be disparate, unconnected observations so far. If you see the connections, you are either ingesting things you shouldn't be or a genius. I don't know which. Questions? Good. I'm glad you're with me. Now, without going through the details of how you do it, what I can tell you is that it is really easy to find the wild-type copy, the normal copy of a mutant gene from yeast, so you can clone that gene with ease. You'll see how you do that later in the lab. So the genes from Pombe and Cerevisiae that correspond to the wild-type version of those mutant genes were cloned. It's very easy to sequence the DNA using the genetic code, which we'll come to later, but you already know about its existence. You can translate that gene sequence into a protein sequence. Now, if you are Jeremy Thorner of the BMB division, you can look at a protein sequence and be able to figure out what that protein is likely to do. Everybody else on the planet does it a different way, which is to use computers and the National Center for Bioinformatics, the NCBI at NIH, to compare your protein sequence against the sequence of every other protein that has ever been sequenced. And the idea is that if it looks like a duck and it quacks like a duck, it's probably a duck. So what does that really mean? Well, it turns out that in the billion years that life has been evolving, proteins have not changed all that much over that period of time. If you were to take two proteins that do the same biochemical function that have been separated by 1 billion years, you would find that they are anywhere from 20% identical in protein sequence to 99% identical. That's really astonishing when you think about it, that you can use a protein sequence from an organism that is 1 billion years diverged from another organism to infer what the function is. In the case of CDC2 and CDC28, it turns out that that protein is about 40% identical to a large family of proteins called protein kinases. So protein kinases are enzymes which put a phosphate group on other proteins. So this is cool. We now have a biochemical clue. We know that there is a protein kinase whose activity is necessary to initiate and drive the cell cycle. Okay? That's kind of cool because we have this autocatalytic thing going on here from Masui, and this is really the first thing that makes us start to appreciate that these different observations in different organisms might be related. Okay. The cyclin proteins, I neglected to say, were sequenced as well. They were sent to the computers at NCBI, and they came back and said, doesn't look like anything to me. All right? But the cyclin protein sequence was then in the database waiting for somebody else to find something that might inform what cyclin does. So we've now got a protein kinase that's driving the cell cycle. We've got an oscillating proteins that we don't know if they're important or not. And the scene now switches to the University of Colorado with a heroic experiment by Jim Mahler. Jim Mahler, at the time, must have had one very big colony of Xenopus, because what he was able to do was to squeeze the abdomens of enough mama Xenopi to be able to fill a 1-liter flask with Xenopus oocytes, each about 1 millimeter across. Okay? Guess what happens next? The blender. So long, frogs. And what he ended up with then was a crude extract of Xenopus. I'm sorry, I forgot to say he had treated this liter of Xenopus with progesterone, right, to activate their cell cycle, blended them, and then decided that what he was going to try to do was to do what Masui was never able to do, which was to biochemically purify MPF. Now, we've talked about protein purification before, a la Kornberg. We discussed before how you use a series of columns to fractionate the extract into different fractions, some of which should have the activity that you're after, and others of which should not. Several of you have asked a very important question I'd like to clear up now, which is in the past, for example, I described a column whose matrix might have a positive charge to it as a way of separating proteins by virtue of their charge. Some people have asked, well, does that mean that a protein either sticks to that column or doesn't stick to that column? So is it either it's like a binary separation? The answer is no. It's more of an analog separation. Proteins are not either charged or uncharged. They are highly charged in a positive way, highly charged in a negative way, or something in between. And any one of these columns simply separates the whole mix of proteins in an extract into fractions that have some common property. So what Mahler does is take this liter of extract, fractionates it over a column, and now he wants to figure out which of these fractions has active MPF. So he needs an assay. What would he do to assay those fractions from that column for MPF activity? What would you do? Any ideas? You need an assay to purify an enzyme. What do you think? What's your name? Greg, what do you think? Yeah, exactly right. So Greg's idea is, well, you take a little bit of this, put it in a syringe, stick it in a naive oocyte, and ask which fraction makes that black hemisphere have a white spot. And that's exactly what he did. So the crude fraction was fractionated. He ended up with an enriched fraction. That was fractionated. Each fraction was tested by injecting it into another oocyte white spot, highly enriched fraction, and eventually he had purified MPF. The purified MPF activity turned out to be a protein of two subunits, a big one and a small one. Now, protein sequencing can be done directly. It's not as easy as gene sequencing. You simply use a gas phase, space age, super femto nano nothing gizmo, and you can take the protein, lose most of it, but hopefully have enough left to actually get you a protein sequence. And then if Thorner's not around, you send it to the NCBI database. And what he discovered is that MPF is a protein with two subunits, one of which is cyclin B, and the other of which is the frog version of CDC2. Okay? Now that's stunning when you think about it, right? Because what we have here is the evidence that the enzyme which drives mitosis in the frog uses a subunit, which is the same as in an invertebrate, a clam, and a protein kinase, which is the same enzyme that yeast genetics found from creatures that were diverged 1 billion years before. So that says that the mechanism for driving the cell cycle of eukaryotes got invented once, and only once. And the most dramatic proof of that came from an experiment that Melanie Lee did when she was in Nurse's lab. And she took the POMB CDC2 TS mutant and simply asked which genes from the human genome, when put into that mutant, could let that mutant grow at the high temperature. She found that gene, she sequenced it, and lo and behold, the sequence of that human gene was about 60% identical to the POMB CDC2 gene. So it's the human form of CDC2. So that's really an astonishing result. So how do we put all of this stuff together now? Well, the way we think about it is that MPF is a version of the cell division kinase, abbreviated CDK, which has one of the cyclin subunits, cyclin B, but not a different cyclin subunit, cyclin A. So here's now a biochemical view of what happens in the cell cycle. In S phase, we have the continuous synthesis of cyclin A, which reaches a peak, and the cyclin A subunit associates with a catalytic subunit of CDK, and as it achieves some threshold, is able to phosphorylate targets in the cell that drive S phase, and then it is destroyed. Boom! Meanwhile, the destruction mechanism that has been chewing up cyclin B is turned down. Cyclin B activity goes up. The cyclin B subunit joins the catalytic subunit that is now liberated from cyclin A, and it then has the capacity of phosphorylating other targets, which drive mitosis. Okay? So the CDK substrate specificity then, crudely speaking, is determined by the cyclin subunit. So our challenge now is to understand what drives this periodic destruction of these subunits. And the answer turns out to be really, really interesting. It turns out that there is a protein that is present in all eukaryotes. It's almost identical, 99% in all eukaryotes, called ubiquitin. It's either a big peptide or a small protein. The choice is yours. It's 76 amino acids long. And I refer to it as the kiss of death, because the ubiquitin protein is actually tagged on to other proteins to cause their destruction. The discovery of ubiquitin and the unique protein chemistry by which ubiquitin is tagged onto other proteins was worth a Nobel Prize for Hershko and Chichinover. And when cyclin A is designed to be degraded, It first achieves one ubiquitin that's ligated onto it, and then more ubiquitins are ligated onto that, so the protein becomes a furball of polyubiquitin onto the cyclin A. This ubiquitination of cyclin A happens here at the peak after the cells have gone into S phase, and the ubiquitination of cyclin B doesn't happen until here. All right, so we've got the proteins ubiquitinated. It's the kiss of death, but how does that work? And the answer turns out to be, again, really interesting. It turns out that polyubiquitination directs proteins to an organelle inside a cell called the proteasome. Now, the proteasome is not an organelle in the sense of something that's wrapped up in its own membrane. It sort of looks like, at atomic level resolution, like an eensy-beensy little garbage disposal. It's got a lid, it's got a cavity, and proteins get inserted into the proteasome, and the button is pushed, and whoosh, they turn into amino acids. Okay? So polyubiquitination of proteins is the signal that causes them to go to the garbage disposal. But now the question is, how do we control when a particular protein becomes polyubiquitinated? And the answer is becoming increasingly known. In fact, one of our colleagues here at Berkeley, Michael Rappa, specializes in the study of the enzymes which put ubiquitin onto substrates. And there are ubiquitination enzymes. There's a bunch of them. I think there's in the neighborhood of 14 different ones in some eukaryotic cells, more in humans. There are many different types, but they seem to have a pattern of consisting of 3 different subunits called E1, E2, and E3. It's not particularly important. But different ubiquitinating enzymes are specific for particular targets. One of these enzymes, called SCF, specifically ubiquitinates cyclin A when cyclin A needs to be ubiquitinated. The anaphase promoting complex ubiquitinates cyclin B. Okay? So we've got quite a ways in our understanding of the cell cycle, but we haven't really completed the circle. We haven't gotten all the way back to boards 1 and 2. Because now the question becomes, how do you control the activity of these ubiquitinating enzymes? And it turns out that the activity of those ubiquitinating enzymes is in fact controlled by the cell cycle checkpoints. So we covered a lot of ground here today, but the big take-home message is that the eukaryotic cell cycle is driven by the cyclic destruction of a cell division kinase regulatory subunit through specific and focused proteolysis. Questions so far? Yes? The question is, how much does a protein need to be ubiquitinated to go to the proteasome? Monoubiquitination, one ubiquitin on a protein, is not enough. Okay? Polyubiquitination is, and the going from 1 to poly seems to be a highly cooperative process. So I couldn't tell you whether it's 2 or 3 or 4 because it's really hard to resolve that. There's a lot of ubiquitins that get added almost in a blur of activity. Yeah. Other questions? Yes?
How are the cells like different from like feedback inhibition? Or are they like similar to that?
I couldn't hear the question.
I'm saying, how are the cell cycle checkpoints similar to or different from like feedback inhibition, like in general?
So, the question is, how are the cell cycle checkpoints different or similar to feedback inhibition? In fact, they're an example of feedback inhibition, right? Not in the enzymatic sense, but in the regulatory sense. What we have here are many cycles of amplification and autocatalysis and cross-regulation because we're dealing here with the control of cell division. And cell division, of course, has to be really tightly controlled, and so there's all these intersecting parts. So I have a question for you then. The part here that ought to make you a little bit puzzled is the CDC28 mutation. How come it's arresting in G1? That should be a puzzle to you. Think about that for a minute. Actually, think about it for 30 seconds. Remember, Masui's dealing with an enzyme that drives mitosis. I'm telling you all this stuff is connected. I'm telling you there's the unity of mechanism driving the eukaryotic cell cycle. But the CDC28 temperature-sensitive mutant arrests in G1. Anybody got an idea for why that might be? Yes, what do you think? The hypothesis was that G0 branches off of G1. That is true, but it's not the explanation. Think about that until Friday. It'll be fun. We'll start Friday's discussion with a special property of that particular mutation and how it all fits